Welcome everyone. So we'll get started with today's grand rounds. Before we begin, I would like to respectfully acknowledge that we're located on Treaty 6 territory, a traditional gathering place for diverse Indigenous peoples, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwa, Saulteaux, Anishinaabe, Inuit, and many others, whose histories, languages, and cultures continue to enrich our vibrant community. So for today's grand rounds, I'm very pleased to introduce Dr. Tabatabaei Dahkili. He started his faculty appointment last year in the Faculty of Pharmacy and Pharmaceutical Sciences. So Dr. Tabatabaei is a medicinal chemist whose research spans stages of preclinical drug discovery from target-focused molecular design and hit identification through lead optimization and preclinical validation. And we think this is really important for us to bring Dr. Tabatabaei to give grand rounds today, because this will be of great interest to not only our clinical faculty, but of course our basic science faculty as well. So for our entire department. And Dr. Tabatabaei, as you'll hear from his talk today, will be heading a unit, a core facility unit provided by the Cardiovascular Research Institute on drug discovery. So that means if you have good target hits or you would like to develop drugs against certain targets, this will soon be a service provided by our faculty. And so we think it's really important to get exposure to this from Dr. Tabatabaei. Okay, thank you.
Thank you very much, Dr. Sutendra. Thank you for the invitation, and thank you for everyone here for attending today. So before I start, I want to declare that there's no conflict of interest or relevant financial disclosure I have today. And I thought I start today with a little puzzle that also very much related to what I'm going to talk about today. I don't know how many of you play chess, but and know the rules. But can you find mate in one? Sorry, white to play. Yeah, yeah, it's for white.
Maybe I don't know what it's called, the one you find number 3. Would that be?
So this one, the pawn.
Yes,
Yeah.
pawn c4.
Pawn c4. It should be immediate mate. So
It's moved a pawn, 2 pawn up 2, right?
No, it was just.
2 c pawn. What else you beat them here.
that's what I was going to tell. Like that's what I'm. So like if you think about it, like a good move here, it's like you have a hanging queen, right? Like you want to just get rid of the queen, right? Like that's a good move. But actually, the winning move here is not the move that you think of, but that's the immediate checkmate, right? And that's what happens in traditional drug discovery. I mean, we usually try to find the best move, but that's not always the checkmate move. But what modern drug discovery works is you evaluate the position, you know how the other pieces are working together, and then you make the move, the winning move. But before I start my talk, I thought maybe I give a little bit of background about myself. I grew up playing a lot of chess before going into being a pharmacist and practice for many years. But I would like to also confess that, as John said, Dr. Sutendra recently beat me in chess. It came as a total surprise. It was not because he played a brilliant chess, it was because it was the night of CIA charge submission. He chose a good night. I was not sleeping for 2 days. So, and I'm a sore loser. That's why I'm talking about this. But yeah, after practicing for a pharmacist for a few days, I decided to follow my dream of doing drug discovery, and I joined I went to University of Salford to do a master's in analytical biosciences and drug design. I was working under Dr. John Hatfield. It was mainly on the synthesis of beta-carbolin derivatives as potential anti-cancer agents. It was there that I was introduced into the science of computational drug design and mostly medicinal chemistry. But since my medicinal chemistry part was lacking, I was really into the chemistry. I joined Faculty of Pharmacy here at the University of Alberta. I joined Dr. Carlos Velasquez, a medicinal chemist, and I did my PhD finding novel targets and novel drugs for a transcription factor called FOXN1. We did a lot of things. We identified the binding site, and we designed a couple of drugs. One of the first few drugs that inhibited this transcription factor. Transcription factor is very important for cancer. And then I joined Dr. John Usher here at the Faculty of Pharmacy as a postdoc for 5 years. I will talk a lot about this in a minute, but basically we worked on a drug discovery of a drug called PSSI51, which inhibit an enzyme called SCOT, which is an important ketone body oxidation enzyme. The drug is basically a novel anti-diabetic drug to help people have their blood glucose under control, at least in preclinical mouse models. But my own lab, I basically started my lab January 2025, and my lab basically focuses on obesity, drug discovery for obesity and type 2 diabetes. And whenever we hear about drug discovery, we are talking about more traditional way of drug discovery, which I need to confess, because most of the drug we're using today has been the result of the traditional drug discovery, which is not a bad thing, it's great. We have to be grateful for having this, but I want to also explain this a little bit more, but super quickly, that in traditional drug discovery, how it works is you usually have a target. It can be an enzyme, it can be a protein, or whatever you want to inhibit or you want to activate, and you start finding a drug that binds to activate or inhibit for it, either through different techniques, either through in vitro screening or in silico screening using computer. And then you have to develop some screening assay for them to confirm them through different assays. And once you have a compound that we call it a hit, which is a drug that not necessarily your best drug, it can be a weak drug or it can be a good drug. You start working on that hit until you keep optimizing that drug to make it more potent. And once you have a compound that is lead, which the lead means a drug that is more potent than your initial finding, you start to optimizing it. Then once you optimize it, and by optimizing, I mean optimizing it in terms of distribution in the body, in terms of decreasing its toxicity, improving its safety, or even potency, you'll have a few lead candidates that you take it to the preclinical testing in different animals, or a combination of those. And then out of those preclinical testing, usually you'll end up having one final lead compound that is ready for clinical trials. But this process usually takes a lot of time, a lot of money. We'll talk about that in a moment. But usually this process is super long and very error prone. And sometimes you have a finding here, you have a lead compound that doesn't work in the preclinical and vice versa. And then you need to do a lot of optimization, takes sometimes 7-8 years. My point here is this is not a bad thing. Actually, my postdoctoral fellowship, I completely followed this. So in my postdoc, John Usher had a target. He found that in obese mice, the activity of an enzyme in the skeletal muscle goes up. It's called the same Scott enzyme. And his idea was if we inhibit this Scott enzyme in the skeletal muscle in obese mice, it can potentially improve glucose control on those mice. So we had a target. So the first step was done, which was the target identification. And this is the real structure of Scott enzyme. It has a nice binding pocket that we can work on. And since most enzyme, we have to do enzymatic reaction, they usually have a nice binding pocket. They call the catalytic pocket of that enzyme. So we have a good pocket to work with. And then for the screening, we use like the computer, like the thing that I used to work in my master's and PhD a lot. And we were lucky that with the first set of screening and the FDA-approved drugs, we found an enzyme, a drug, FDA-approved drug called pimozide as our hit. This pimozide, I'm sure most of you know, is an FDA-approved drug that blocks dopamine receptor in the brain. It's initially they made it for schizophrenia, but currently we are using it for the Tourette syndrome to reduce tics. When they tested this drug, in the process of drug discovery, we call it the screening. It worked by reducing the activity of Scott enzyme in the muscle, and it also improved glucose tolerance. But since it's a hit compound, this pimozide is what we call it is a dirty drug. But by that, it means that since it's an antipsychotic, it goes to the brain to inhibit dopamine receptors. That's why it's a drug for schizophrenia and psychosis. The other problem with this drug was all the side effects of these actions. When it gets into the brain, it causes a lot of other symptoms that we call extrapyramidal symptoms as a result of dopamine antagonistic activity, which causes like the muscle spasm, stiffness, and tremor. The other problem with the pimozide was pimozides are ketone body oxidation inhibitors, as we found. And when they get into the brain, they also inhibit ketone oxidation in the brain. And we know that ketones are essential fuel for the brain. So we didn't want this to happen. So one of the biggest challenges was to reduce the penetration of pimozide into the brain. Here is the idea that we need to optimize our lead compounds to make it less brain penetrant. And that was part of my postdoctoral fellowship. And in the process of lead optimization, in a less classical way, but a more modern way, you have a pharmacophore model. And in a pharmacophore model, we know that around the molecule, what are we looking for, and what are the essential features of that molecule that we want to make. And we were lucky that we have a hit, which was a pimozide. We knew how it binds to the receptor. So we started doing the hit generations, and we had to filter a lot of compounds based on 2 criteria. And our criteria was you should have limited brain penetration and it should not block the dopamine receptor. And we ended up synthesizing a lot of drugs, almost 55, 60 drugs. And out of them, a compound called PSSI51, which we call it preferred selective Scott inhibitor 51, was emerged as our most promising compound. And then we showed that this drug actually binds very similar to the pimozide to the receptor, which is the Scott enzymes. When we tested this drug in a Scott activity assay, which is still this process we call lead optimization, now we are screening our lead compound, it inhibited the Scott activity. And when we tested this on this artificial blood-brain barrier membrane, we found that it doesn't get into the brain, at least in artificial membrane, which is the Pampa assay. The second criteria was it shouldn't inhibit the dopamine receptor like pimozide. We did a few tests and we confirmed that it does not touch the dopamine receptor. But how about its activity in preclinical models? When we put mice on high-fat diet for 8 weeks and we gouaged 51 and the pimozide together, not together, but I mean together like different mice, of course, we found that they have very equal potency on glucose tolerance. But how about its safety? When we put mice on voluntary vills and we gouged both pimozide and 51 together, we found that pimozide mice are super docile, which is very likely due to all the extrapyramidal side effects. They didn't want to run, their run speed was low, they were not very active animals. In comparison to our pimozide mice, our PSSI51 mice are very active, no at least activity-related side effect, which was expecting because it doesn't get into the brain and it doesn't inhibit the dopamine receptors. We also did a mass spec of the brain, and we found that a significantly less PSSI51 gets into the brain, which was very exciting. And this work was published last year and resulted in the development of first-in-class, the brain not penetrating ketone body oxidase or a SCOT inhibitor that inhibit the ketone oxidation peripherally and improve glucose tolerance, which we think it's a new way and it's a new class can serve as a new class of antidiabetic drugs. What I just told you today was like a very traditional way of maybe finding a drug, which was, as I previously mentioned, finding a target, of course, and just screening a lot of drugs and then synthesizing a lot of drugs and test them all and then keep going to the preclinical testing. Although we were very lucky that we found pimozide as our target, that we can quickly optimize it and get into a good drug, which was the PSSI51. But usually this process, sometimes it's a little bit harder than we think, and it's not so straightforward, especially for the drugs that don't have like a defined binding site. And this is a good example of the project I did for the person I lost the chess match to, Dr. Satendra. When I was still a postdoc, he approached the lab and he was working on a transcription factor called zinc finger protein 281 or ZNF protein, which is a transcription factor which is regularly or frequently overexpressed in various human cancer. The problem with this zinc finger protein was, if you look around it, like there was no specific binding site for this protein, which is a very a normal feature of transcription factors. They don't have a definite pocket, of course. And what we wanted to do with this is we initially wanted to develop, I was thinking about it, to develop an in vitro screening assay for it, but making this zinc finger protein was difficult. Developing an in vitro assay, although possible, there are so many ways of developing high throughput assay for these zinc finger proteins, but also very difficult and expensive, and optimizations are definitely very difficult. So we thought that, okay, there's no binding pocket, but we know that this transcription factor binds to the DNA. Let's try to see if when it binds to the DNA, there is a binding pocket that forms, maybe it changes the structure. And when we did that, And when we did that, and we usually do it with a method, a physical-based method called MD simulation, which is a way of seeing the proteins not in static ways, more like a real-life movie. When we did that, we found that this transcription factor, ZNF2A1, it will form a very transient binding site at the interface of the protein and the DNA. And then we perform computer modeling on those transient binding sites. It was not just one transient binding site, it was developing multiple ones. And we have to do a lot of computer-based modeling and screening. And we finally found a good binding site, and then we use computer to screen a lot of drugs, designed a lot of compound, and find a drug that we call it the ZNF281 interfering molecule, or Z. This drug was basically, after we did the MD simulation, we found that it was interfering with the binding of the ZNF to its to the DNA, and it was just detaching the DNA and protein. When we synthesize it, I don't talk about all the details, Dr. Satendra will have his paper out soon, but it was working both in vitro and in vivo, and was able to shrink down the tumor size, as you can see here. And this is a good example when the more traditional drug discovery may stall at some point when we don't have a good in vitro screening assays, and we don't have a definitive pocket to work with. But in my own lab, my lab works on obesity and type 2 diabetes. And one of the areas that I am working on are on incretins. And I'm sure, like most people now know that, like Ozempic, the semaglutide injection, they work by mimicking the natural incretin, GLP-1, which enhances insulin release and just reducing appetite. It is revolutionizing the treatment for diabetes and obesity. And even the newer ones like Mounjaro, tirzepatide, which is a GLP-1 GIP receptor dual agonist, and which they have a better efficacy, which they think they have fewer side effects. And then even the newer ones, which are the triple Gs, like the GLP-1 GIP and glucagon receptor agonists, which the goal was to have even larger fat loss and preserve the muscle loss related to the weight loss. But the problem with all these drugs, other than all the side effects, of course, was they are injectables. They have very short shelf life. Almost after they open it, they have like 50 days to 56 days, I guess, to inject it. And they are, of course, very expensive. The pen itself is very expensive to manufacture. The drug peptide itself is expensive to make. And at the end of the day, it's expensive for the patients to take these drugs. So the scientists in Big Pharma tried to solve this problem by adding an absorption enhancer called the SNAC to the peptide semaglutide. And how the SNAC works is, in presence of a SNAC, it increases the gastric pH and increases the epithelial permeability, and it lead a little bit of the peptide to get into the circulation without being denatured. But still, the bioavailability is very low, 0.4 to 1%. If you compare it to the actual injection of semaglutide, which is 89% bioavailability, it's almost nothing. In the clinical setting, clinical trial, 14 milligrams of Rybelsus is equal to 0.5 milligrams of Ozempic in terms of weight loss, still working, but it needs strict fasting condition. If a patient doesn't follow all those fasting conditions, it has a very variation daily in terms of the drug exposure. The other problem with this is the long-term consequences of this daily gastric microenvironment disruption is not known. We don't know, while the snack is very safe, we don't know what happens if we disrupt it every day. And it's still a peptide. So you need to spend a lot more money making the peptide in terms of volume. 14 milligrams in each pill is still a lot, and it costs the same. So we're still not there. So the better alternative would be a small molecule, which is orally available, it's a longer shelf life, more predictable PK, and of course, lower cost. The problem with these making small molecule agonists for this receptor, they are from a class of G protein coupled receptor, GPCR, is they belong to the BGPCR class. Most of the drugs we have today are from the A class GPCR, like the beta blockers, antihistamines. They are meant to recognize smaller hormones or smaller ligands. But this B class, they are meant to identify and activate by long peptides. And that makes them very difficult to develop a small molecule for them. And how it works in real life is, in the BGPCRs, they usually have an extracellular domain, ECD, and a transmembrane core. Initially, a peptide needs to be recognized by the receptors. And once it's recognized, it just docks it into, if it's a right peptide, it docks into the receptors and starts signaling. And for this GPCR signaling of this GLP-1 receptor, for example, they can signal through two channels. One is the G protein couple, like protein G, GS, which is responsible for all the metabolic effects. They can also signal through beta-arrestin, which they think causes all side effects. When you are working with the peptides, it's a little bit difficult to bias toward the GS or the beta-arrestin. But when you are working with small molecules, you have more knobs to turn toward one of these directions. When I say it's difficult, I didn't say it's impossible. Of course, Eli Lilly made one, Pfizer made one, which failed, but Eli Lilly has this drug called orforglipron, which is actually a GS biased partial agonist of the GLP-1. It's doing great. Phase 3 clinical trials, 10 kilogram weight loss, even at 72 weeks, 12.4% weight loss. It's doing awesome. But for me, I can't compete with Lilly. We need to do to go to the next receptor, which is the GIP receptors. And it's shown that inhibition of the GIP on itself is very helpful for obesity and type 2 diabetes. So in order to make the first ever small molecule of GIPR agonist, I had to come up with a novel solution. What we did was, we still did the molecular dynamic simulation, which now it's a way of how, for example, in terms of the GIPR, how the real world movie is. How does the peptide affect the backbone of the receptors? And what are the consequences of this peptide on the backbone of the receptors? And basically, what I called it, the dynamic interaction fingerprints will record all the transient contact and the state that predicts agonism. So what does the peptide do to the receptors at the molecular level? And if we can learn from those, if we design molecules that mimic these, we can basically synthesize them and get a small molecule agonist of the GIP. And we did that. So basically, we developed an AI platform that can learn from all these transient contacts and start generating molecules that can mimic all those contacts and synthesize and tell us what is the best molecule that can mimic those contacts. So probably that small molecule will be the agonist of GIPR. And once we did that, once I developed that, I was not really believing in that I can do it. But the first molecule that this platform gave us, it gave us basically 5 drugs to make that can mimic GIP. And drug number 3, once we synthesize it, was working. And the good thing about AI is you don't know how it is doing it because it's learning based on all those small details that you don't know. So basically, we came up with this drug molecule. We call it G3, which was equipotent, at least in vitro, and we recently tested it in vivo in islet perfusion. That is equipotent to the GIP peptide. And this is super tiny molecules. And it is amazing that such a super tiny molecule can act exactly like a big peptide. But to my surprise, like 2 weeks ago, Eli Lilly published a patent of the GIPR agonists. While my heart sank at that moment, but the thing I want to talk about today is if you look at this molecule, they made over 200 compounds. For a chemist like me, making this is definitely a nightmare. They probably spend a ton of money optimizing this. You can easily tell by looking at the deuterium that they put there. It means that they had a lot of issue there, that they put deuterium there. But for us, it was just me that I made 5 compounds in total. My other lab tech, it was at the time for John lab tech, we bought a cell line for the GIP for $1,000 from UBC, and then we tested our drug, and then we have it. It took us like 1.5 months. So I just wanted to see the differences, right? So probably we spent like millions of dollars on that. We spent like a couple $1,000 to get where they are. And that's very important for us because that gives me that boost of, instead of being so sad about this, it gives me a little bit of boost of self-confidence that other things are also possible in this platform. And one of the other things I'm doing is, if the platform can find, based on the dynamic interaction fingerprint for 1 receptor, why not for 2 receptors? So it will be a small molecule equivalent of tirzepatide, which is Mounjaro, and we did that. And like last week, for all these molecules that we are making, and like we have very promising results of a dual small molecule agonist of GIPR and GLP-1. And why not a small molecule equivalent of retatrutide that can activate 3 receptors? And we are working on that. And that opens up the door for the polypharmacology of the things. For example, in cancer, in many diseases, you need to activate 2 receptors at the same time. So why not developing a drug that can activate or inhibit 2, 3, 4 things at the same time under your control? And that is where my research at some point will go to develop more drugs based on the thing I am developing right now, which is at this point working really good and precise. But still, this process of dynamic interaction fingerprints still is a not super fast process in terms of drug discovery for your targets, because you still have to synthesize a bunch of drugs, although it's very precise. And at some point, you have to synthesize drugs that are very difficult to make. For example, for the triple agonist, we have a candidate that is, we are trying for like 2 months to come up with the first step, which is not possible. So we have to keep working on it, which is not super fast and easy at some points. But how to boost the speed? So through a GELF application, I'm getting a device called SPR, which is a surface plasma resonance. This is the newer version, which we call it SPR 64. And before talking about it, let me tell you a little bit about it. Probably, I guess the only person knows is Peter here. So how the SPR works is at the bottom, there is a glass prism coated usually with gold. On the surface, depending on the protein we want to load, is the surface linker. What you need to do is basically we coat the surface of the gold with the protein of the interest, the target that we want to find drugs with. And once we have that, we start to shining a light here to the prism. And this surface is very sensitive to changing the mass on the surface. So when we add our drugs or whatever analyte, drug, protein, whatever we want to find for that binds to that protein or target, it will change the reflection of the light from the prism. And then we can record that, and that's somehow proportional to the amount of analyte bound to the protein. That's basically a very fast and quick version of how SPR works. But in terms of boosting the speed, this 64 SPR is not a conventional SPR. It has 64 channels. So instead of having one of those sensors, we have 8 and 8 channels. It means that in theory, you can screen 8 different targets with 8 different drugs, or one drug at 8 different concentration. It has a screening capacity go, no go. And by that go, no go means a drug is binding or not binding, and 10,000 drugs per day in most realistic condition. But thanks to the new CFI that we are putting forward with the CVRI, we are even maximizing the throughput to even 200,000 drugs per day. And how it works is through the liquid handler called ECHO. So basically what ECHO does is it will pool the drug in each plate for us. So instead of having one drug per plate per well, we will have 50 to 100 drugs per plate per well, which will increase the throughput for us. But the thing about the SPR is not about the speed. It's about the information it will give us to put into our platform to learn. Once we gather all this information, how much a drug is working, if it's for a target with a specific binding site, with a specific amino acid 3D shape, how does that shape, how does the amino acid are interacting at the molecular level, and how deep is that binding site? What is the shape of that binding site? Is that drug that is binding that to the target, is it a weak binder, a strong binder? What is the KD? What is the K on and K off? All those information going into our platform. At some point, it will learn a lot from all these throughout the years. That theoretically, at some point, it should give us a drug just by looking at the binding site precisely, or at least, I don't want to make a big claim, but at least it will reduce the time or the number of small molecules that uh, we need to screen using the SPR or other assay. So basically, it will increase our chances, reduces the speed, and also reduces the cost of drug discovery, preclinical drug discovery, hit to lead identification. Thank you very much. I would like to thank everyone. Mostly these people helped with the postdoc fellowship project PSSI 51, and thank you very much.
Okay, thank you, Amir. So we can take questions either in the audience or in our chat. So will you read them a little bit?
Yeah.
Oh. Okay, so the question is three unrelated questions. So the first is, do people vary in their sensitivity to GLP-1R and GIPR agonists, or would all respond to GIPR?
Do you want me to?
Yeah, sure. Why don't you answer that first then?
Yeah, I mean, there is a I really don't know the answer to that, but there is a preprint saying that people have different people have when given GLP-1 and GLP-1 agonists, they have different response to, but they have different bias response in different tissues. So that's very possible. GIPR agonists also have anti-inflammatory effect. I don't have the answer to that. Are these small molecule easier, cheaper to synthesize at scale? Of course, they are easier. Like one of the things that I'm planning to do, and part of this platform is we will also screen and prioritize all those drugs that are easier to synthesize. So one of the factors is, can we synthesize them or we cannot synthesize them? And for example, for the small molecule GIPR we have, we made it in one single step. So that's a very important consideration when developing a small molecule. At the same time, we want to make a small molecule, but we don't want to spend 2 years making small molecules like the one Lily did. Yes.
Your first project, you screened a lot of FDA-approved drugs. How do you see that changing? I can see the efficiency perspective that we'll be able to screen in different ways or even design de novo after there's learning through this.
Yeah, and of course, you can learn from those things too. But for an AI platform, you usually need a super large library of things to learn from, mostly from experimental validation. Yeah, possible. Yeah, I have to just go back and do all these things for sure. But what my platform is doing is it is now learning from all the things we have in the literature, and so I try to learn things as we go. But we have to also be very careful to do supervised learning, not to learn everything that is there, because you can't trust everything. And once you have something that is not real, then you are in a big danger zone there.
Okay, we have a question online. So nature likes to reuse protein structures for different things. So it shouldn't be surprising that drugs have off-target interactions, particularly when you try to target more than one target. How do you deal with that?
That's a very good question. So mostly small molecules are predicted to have, even the drugs that we are dealing today, probably to have 200 plus of targets, although it's very dose dependent. Of course, we will have that with our small molecules. There are ways that we can predict that. The machine learning world is improving significantly. So basically, you can also predict, at least for important targets, if they can be they can have off-target effect for the top 50 proteins using machine learning. So it's possible to somehow reduce the likelihood of off-target interaction with small molecules, and that's one of the biggest problems with small molecules. But always you need to do screening at some point. There are so many panels for toxicity screening that we can do, and when you do all those screening, the good thing is our platform can learn from those things, and it will design drugs with in the next round that doesn't have all those features. But at the end of the day, you need to still screen for toxicity, and most of these drugs, when they go to preclinical testing, they need to get through all those screening and assay for toxicity. And yeah, this is a good problem. It's a big problem for small molecules, but there are ways to overcome that for sure.
So I was a little puzzled by your emphasis on finding a single agent that could target more than one receptor. It seems like the task there kind of scales geometrically, whereas if you've got two small molecules, one for this receptor, one for that receptor, that probably scales arithmetically. And so sort of why do you go to the extra work of trying to have one small molecule to target two things and just one power pill with two things in it?
Yeah, no, you're absolutely right. But the problem with giving 2 or 3 drugs at the same time is now you are dealing with 2 or 3 drugs with different PK, where the system or the body will see these drugs at 3 different times and not at the same times. Are they also working against each other, working at the same time? And that's the basic reason behind now big companies spending so much time in making like a dual agonist or triple agonist for these targets. So basically, when you have 3 targets, your body, the tissue of target, we won't be able to see those targets at the same time. So you have different PKs, you have different response. And they have shown this before, that having single molecules, dual targets, superior response for sure.
Interesting, thank you.
All right.
Yeah, great talk. Just wondering about like all of these GPCR agonists. You kind of have them modeled with the peptide going transmembrane. Usually that means that it's like super hydrophobic. Do the therapeutic peptides have like a transmembrane helix? And I mean, if you try modeling Eli Lilly's compound, does it find the transmembrane segment, or is it mainly extracellular?
Yeah, like we have no crystal structure in Eli Lilly compound yet. I think they have hard time doing that.
It's hidden, right?
Yeah.
Data.
Yeah, and I didn't have even for my compounds, to be honest, I did, but so as you said, like we don't know. Like at least for my compounds, the way I put it, it's more like toward the extracellular domain they will sit. And I did model a lot of Eli Lilly. That's what I think they will see toward extracellular domain and not transmembrane core. Like that's very deep. That's probably not possible for the small molecule to go that deep.
Okay. And do you have why would you deuterate a methyl? No idea why.
Yeah, I think they have solubility. They had solubility issues at the Eli Lilly. So I was reading about like comments on LinkedIn that when they have usually when they have solubility issue with probably they had methyl trifluoro there, and their compound was super insoluble. So they had to just switch it to deuterium to just keep the interaction there, but to still, like in terms of the bulkiness there, but still have the fluorine group mimicking fluorine group there, but improving the solubility.
But the deuterium.
Yeah, probably very similar to hydrogen. Yeah, probably they had other issues. Like I really don't know, but when you see they are deep into adding deuterium, you really don't know what's going on. Like they had really PK issue. But what I know is deuterium, in terms of like PK, they showed that they have an improved PK profile when they have deuterium there. Like I know when they, for example, when they have issues with like substitution of the ring with like hydrogens, which go under, for example, when you have a benzene ring, it goes under like oxidation ring, like, but when you have deuterium, we don't have that, right? So they probably have this PK issue at some point too. That's why I think they had a lot of issue. They synthesized a lot of drugs and they came out, okay, let's do it deuterium, and maybe they solved the problem. Although in their patent, they didn't, they just disclosed in vitro results. They didn't disclose any in vivo results, any PK results yet, but we'll wait for that and see how it goes.
Great talk here. Before I ask my question, I just wanted to add to your response with the single molecule, the 3 targets. And it's all in addition to the 3 different PKs, it's also 3 different compounds that have to get approved, right? Like if nobody can do one compound, it's like way easier for the company. But what I was curious about is with the when you show like you talk about the bioavailability of Rybelsus. So is Rybelsus technically then, because it's like 50 milligrams once per day versus some injectables once weekly 2.4, is Rybelsus then more expensive for both Novo to make and for the patient to take?
It's exactly the same price, like as long as I know, it's the same price, like $500 per pill, like per thing per, I think, 30 pill.
Every day, right? So
Yeah, you have to take pill every day.
does it end up actually being more expensive?
Oh, I actually didn't calculate that. So
It seems like actually it's not.
Yeah.
It doesn't seem like it actually.
So, but I think you like if you have like Ozempic 4 milligram pen, you finish it in 1 week, one month, right? So four injection per month, and then with Rybelsus, I think each comes at 30 pills, 500. So I think it's very similar in terms of cost. I think
the company have to make so much more of the actual peptide because it's so poorly.
Yeah, I think also, as we know, like peptide is very hard to make for the Eli Lilly or the Novo, but the pens are also very expensive. So that's why their costs are similar. Now they are making more peptide for Rybelsus, but they don't have to make the pens. But Ozempic, they have to make the less peptide, but more pens.
Two kind of speculative questions. So you read a lot in the lay press about how drug discovery is going to accelerate, and you're going to really show a nice real world example of that. So congratulations to you and Keith. But what is this going to have from a policy perspective? If you're accelerating and all these other folks are accelerating, what's going to happen, or what do you think is going to happen from the regulatory perspective, healthcare system perspective, and then the cost perspective? And I was kind of curious when you kind of said you make these drugs cheaper. I'm not convinced that the actual discovery and cost of the drug has a direct line of sight to the price that's charged to.
Yeah, I agree. Like, but I hope that at some point, if like the cost of drug discovery, reduce the cost of drug discovery, or we scale down drug discovery to smaller spin-off companies, maybe we can reduce the cost to the patient and the healthcare system. So we know that like big companies, they usually spend like billions of dollars developing drugs. They want to like at least a cost recovery option to do that. But that's what at least we hope to reduce the cost. That's what I hope that one day super fast drug discovery will at least reduce the cost for the preclinical drug discovery. So it translates in the long run to the patient healthcare system.
Okay, we'll take a couple of questions here online, and then so one is there a different environmental impact from pills versus syringes?
Um,
But yeah.
yeah, it's a good, yeah. I don't, I don't know the answer to that. I'm sorry.
Any thoughts on speeding up the regulatory pathway?
Like at my research, I'm doing a lot of preclinical chemistry peptide synthesis now, but I'm very naive into a regulatory pathway these days. I'm trying to get into those sciences now. I'll probably update you next time if I. So yeah, but I know like it takes a ton of time getting these things approved. But yeah.
Okay, I think with that, we'll conclude today's session. So just thank you very much, Amir, for great talk and a lot of interest, definitely. And we look forward to the core coming up, and I'm sure you'll get a lot of clients for the core here. All right, thanks everyone. Thanks, Amir.